Hi everyone, welcome to the development of the urinary system, part 2. At this point, we've talked about how we've built a kidney with an excretory component and a collecting component. So what's left? Well, it turns out that the kidneys need to be in the abdomen below the adrenal glands. And right now, they're not. So we need to move them. We also need to form a separate bladder to collect urine with ureters that connect to it. So let's start off by talking about kidney relocation. So as I mentioned, now we have kidneys, and you can see them here in light pink in this cartoon on the left, but they're not in the right place. So how in the world do they get from here to here? Well, it turns out that this ascent of the kidneys is not an active migration. But the kidneys do relocate from the sacral to lumbar position, and they do this at the same time that everything else is developing, including we should be roughly in the mesonephros at about this time. So how actually do the kidneys relocate? Well, they're going to follow a path just on either side of the dorsal aorta, and as they ascend, they're going to rotate 90 degrees. So they'll start off facing ventrally, and then they will be facing medially in the adult. How does this happen? Well, it's really the result of differing growths of the sacral and lumbar regions of the embryo. So that really causes this unfolding of the lower pole of the embryonic body, and eventually that allows organs to take their place below the adrenal glands. How exactly does this happen is really not clear. So here we can see the kidneys are ascending, and as they ascend, we know that they need to maintain a blood supply. And so they are progressively revascularized by sprouts from the dorsal aorta. And then as those sprouts vascularize the kidney, the others will degenerate. So again, I just want to reiterate that this mechanism is really not well understood. And what's important is that you know that the kidneys have to ascend to their final place below the adrenal glands. Now, of course, like any other process in development, we can get anomalies from variations in this relocation process. Most of the time, these are really truly considered variations because they don't have any phenotypic or functional effect. So here's an example of some accessory or supernumerary renal vessels. So in this cartoon on the left, you can see two renal arteries for this kidney, and then in the image here on the right, you can also see multiple renal arteries for these kidneys. So the supernumerary vessels are really the persistence of any vessels that form during kidney ascent, and that can also include the formation of multiple renal veins. And as I mentioned, most of the time, this doesn't cause any problems. We often see these in cadavers as incidental findings. But sometimes they can compress the ureter. So here on the left, you see a cartoon showing multiple renal arteries, and the lower one is compressing the ureter. And what this results in is something called congenital hydronephrosis. So there are different kinds of hydronephrosis, but this one, again, is congenital. And so this ultimately causes a distension of the renal pelvis and calyces. Now on the right, I'm showing this one type of grading system. And obviously, I don't want you to memorize this. I just really think it's useful to look at the different stages and to see what hydronephrosis looks like both in a cartoon and then also on ultrasound. And what you can see is that if you get to something like grade 4, where you have this extreme dilation, you really lose the kidney medulla and you lose some of the kidney cortex, or at least it's very thin. And so you can definitely see that this would severely impact kidney function. Another example of an anomaly is the ectopic kidney. So most of the time, these ectopic kidneys are found in the pelvis. Sometimes they're just called pelvic kidney. So you can see here on the cartoon on the left, you have a kidney that failed to ascend and also is malrotated. And then you can see in the image on the right, again, the normal kidney on the left side of the body and a malrotated kidney on the right. Now, I did want to point out that in this case, the adrenal gland is located normally, and that's because the adrenal glands don't actually form as part of the urinary system. Here's another example of a type of ectopic kidney. This is a discoid or pancake kidney, and it's usually a single fused kidney. Oftentimes these are linked to mutations in the gene encoding sonic hedgehog. And this gene, perturbation of this gene in particular, often causes things to have midline defects. So, for example, if you perturbate this in development of the brain, you can have a fused cerebral cortex. If you perturb it in development of the digits, you can have mirror image duplications of digits. And we'll talk about some of these as we talk about the development of other systems. Finally, we can have something called a horseshoe kidney. So this is actually pretty common. The incidence is about 1 in 400 live births. You can see a cartoon on the left and a CT on the right. This kidney is usually functional, and it's formed by fusion of the caudal poles of the kidney. Now, because they're now connected, they can't ascend. So that ascent is actually blocked by the inferior mesenteric artery, which you can see here in the CT scan on the right. So what about the adrenal glands? Well, the adrenal glands are formed and then sit just above the kidneys. Supra means above. So how does this happen? Turns out the adrenal glands have a dual origin. The cortex is formed by mesothelium very early in development, and then the medulla is going to arise from neural crest cells. As development proceeds, we have this second wave of mesoderm that comes in, so now the cortex has 2 layers, and this continues even after birth, where the 3rd layer will develop until we finally arrive at our triple-layered cortex of the adrenal gland, along with the neural crest-derived medulla cells that produce epinephrine and norepinephrine. Now again, take-home message here that these form separately from the kidney, they have a dual origin, and that they become opposed to the kidneys as a consequence of kidney ascent. So in those examples that I just showed you where we saw ectopic kidney, the adrenal gland would be in the normal position on that side of the body. All right, let's finish up by talking a little bit about formation of the bladder and the ureters. So the development of the urinary tract is happening at about the same time as the kidneys are developing and as they're undergoing ascension. So again, all of this stuff is happening at roughly the same time. So here's a cartoon showing sort of lower half of the body here, lower part of the embryo, and it's a side view. And what I want to point out is this structure called the cloaca. So the cloaca is this large cavity, and it's a transient structure lined with endoderm. And it's continuous with the early digestive tube that you see here in green. And it's also continuous with the urogenital sinus, as well as this structure called the allantois. So what is the allantois? Why am I talking about it? It is a thin endodermal diverticulum, and it extends into that connecting umbilical stalk. It has 2 parts, an extraembryonic and interembryonic part. The extraembryonic part just pretty much degenerates. But from the wall of the allantois, we see the umbilical circulatory arteries and veins that supply this placenta arising. Now, in spite of this, it's still pretty much considered a vestigial structure in humans, although in lower vertebrates it's important for removal of waste. Now, the intraembryonic part pretty much stays small. You'll see sort of a cord of endoderm that's embedded in the umbilical cord. But there is a part that involutes and persists as a stalk or a thick tube that's called the urachus. And I'm going to come back to the urachus in just a minute. But for now, let's go back and talk about the cloaca. So we have this large area, and we need to divide it into several parts. So how does that happen? Well, it turns out that there is growth of tissue that's called the urorectal septum. And this tissue is going to essentially grow down in a very similar way that we saw the septum of the atria forming. It's going to grow down, and it's going to partition this urogenital sinus, seen in orange and yellow on the left, from the rectum, seen in green on the right. Now, I've shown this particular process in a typical XY embryo, but the process is very similar for typical XX folks as well. At the end, what we see is that that urorectal septum has, again, divided that transient cloaca into multiple parts. We have the developing bladder in yellow, and we have the developing rectum in green. Now, the urogenital sinus itself is going to form the prostatic and penile urethra and the urethra and lower vagina, and then the tip of the sinus is going to form the perineal body. Now, what I want to do is go back and focus on this second image. So originally, as I said, the bladder or the urogenital sinus is continuous with the allantois, and that allantois is going to involute and form a structure called the urachus. And that is essentially a thin tube that connects the outside of the body with the developing bladder. Now, the urachus is going to remove nitrogenous waste from the fetal bladder, so it does have a sort of a function in the embryo. But that tube, like many tubes, is going to eventually involute and degenerate. Now remember, anytime you have something that involutes or degenerates and goes away, in the adult, it typically remains as a ligament. And that's true here too. The urachus becomes the median umbilical ligament. This is a really common theme in development. Now I want you to think back to what we saw in the circulatory system and think about what happens when structures that should close or involute remain open or patent. So this means we're going to have an abnormal connection between two things that shouldn't be connected. So our example would be the patent ductus arteriosus, where we're now continuing to connect the pulmonary artery and the aorta. And so this results in deoxygenated blood moving into the body. Now, the same thing happens here with the urachus, where it fails to involute, and that results in a patent urachus. But instead of blood or deoxygenated blood moving to the wrong place, we can often have urine moving to the wrong place. So there are several forms of a patent urachus. The first is sort of the stereotypical patent urachus or urachal fistula. And this is where we have a complete failure of the urachus to obliterate. And so what we see is a communication between the bladder and the umbilicus. Now, when the bladder is connected to the outside of the body, what happens? Well, what happens is we tend to see leakage of urine through the umbilicus. This is one of the first signs that is seen. Patients with this can also have redness around the umbilicus, abdominal pain, or even urinary tract infection. Another form is a sinus, and a sinus is a pouch that has one open end. So in this case, I'm showing an opening to the umbilicus, but not the full opening to the bladder. We can also see cysts, and cysts are just fluid-filled cavities. Now, neither of these is going to result in leakage of urine from the umbilical cord, but there can be infection, and in rare cases, there is the potential for malignant transformation. All right, let's talk a little bit more about development of the ureter. Now, I want you to recall that the ureters are outgrowths from that ureteric bud, which is an outgrowth off of the mesonephric ducts. Now, eventually, the ureter is going to need an entrance into the bladder. So how exactly does this happen? Turns out that the mesonephric ducts are going to help form part of the bladder, something called the trigone that you can see in gross anatomy, and it will also contribute to the genital ducts and glands in typical XY males, as I talked about previously. But again, the ureter has its own entrance. So this is the classic view of how the ureter achieves that separate entrance. And the classic view really is that the bladder comes from the urogenital sinus, which is endoderm, and the trigone of the bladder comes from the mesonephric ducts. And I'll explain why that might not be true in just a minute. But let's go over this view because it's still tested on the boards. And so I want to make sure that you have an idea of how it occurs. So essentially, the caudal parts of the mesonephric ducts are going to grow, and they're going to be absorbed into the bladder wall. And it turns out that they grow faster than the cranial ends. But meanwhile, the ureter is attached to the kidney, and the kidneys are undergoing ascent. And as they ascend, the traction exerted by the kidneys is going to cause the ureter orifices to move up, and so that they eventually are going to enter the base of the bladder. Meanwhile, those mesonephric ducts are going to open into the pelvic urethra just below the neck of the bladder. And this is called the trigon. It looks like a triangle in the adult. So this is the classic view of how the ureter achieves a separate opening. The newer view is that the mesonephric ducts contribute very early on to the formation of the bladder, but later all of the bladder is overgrown by endoderm so that it completely arises from endoderm. And we base that information on actually analysis of a bunch of different congenital anomalies. All right, now you've probably figured out what comes next. What happens if this process goes wrong? Of course, there are some anomalies of the urinary tract. So one of these is complete duplication of the ureter. So in this case, we really think this happens from the process of duplication of the bud. Or it could happen from early splitting of the bud before they actually reach the mesonephric mesenchyme. Whatever the process, essentially you have the formation of two complete ureters that are coming from different parts of the kidney generally, and they enter the bladder separately. So there are two entrances, and you can see this over in the CT scan on the right and in the cartoons in the middle. Now here's what it looks like in a gross specimen. Now, we can also have incomplete duplication of the ureter, and that really results from a bifurcated ureteric bud. Okay, so in this case, you still have two entrances in the kidney of the ureter, but then you essentially get a Y shape. So the ureters join before they reach the bladder. So by the time they reach the bladder, there's only one entrance to the bladder. You also can see something called ectopic ureter. So here's our cartoon again showing the mesonephric ducts in the bladder and the ureteric bud. And this bud is kind of caudal, so it's reasonably caudal. And what happens to that bud is it's going to induce the formation of the bottom half or the caudal part of the kidney, and then it's going to drain it into the bladder as it would do normally. But we also have an ectopic bud, a cranial bud, and that bud induces formation of the cranial part of the kidney, and then it gets carried down with the mesonephric duct. So it doesn't stay with the caudal ureter. So it looks something like this. So here you can see a kidney, and in yellow you see the normal ureter that is coming from the kidney and emptying into the bladder. And so we see urine coming out of the urethra. But we also see this ectopic ureter in orange that forms a ureter that has abnormal entrances. So where are those abnormal entrances? So it can enter into the vagina or the vestibule or the urethra. And again, I'm showing this in a typical XX person. And so this sign that we would see here in this patient, for example, might be dribbling of urine. So you can see that urine would be coming out of the vagina as well as the urethra, or it could be coming out of the vestibule. Well, that's it for this video, but I want to recommend that you watch this other video from Medical Animations. So this is the development of the urinary tract. It's about 6 minutes long, and I think it's a really useful video because it shows the ascent of the kidneys and it shows how the ureter achieves a separate entrance in animation. And I think it makes it a little easier to grasp how those processes might be happening. I really think it's worth the 6 minutes, but if I were you, I would turn off the groovy music.